thank you for the invitation to speak. I've really enjoyed the presentation so far. I changed slightly the title of my talk and to follow on Wendy's presentation. Function actually is really where things are really important, and we really need to elucidate the function by which the microbiome is actually affecting the host if we want to be able to modulate the microbiota to affect health. And so the title of my talk here is Functional Insight into the Role of the Vaginal Microbiome in Sexually Transmitted Infection. Just before, I have some disclosure. So the vaginal microbiota in reproductive age women is as opposed to the gut, is dominated mostly by one species of bacteria, Lactobacillus, one genus of bacteria, Lactobacillus, and represented by several species of that genus. And that's why we considered an optimal vaginal microbiota. So alpha diversity is very, very low. Lactobacillus is thought to be beneficial because it produces lactic acid and it can acidify the vagina to pH 4. And you can think about many ways this can affect protection against different conditions, including infectious disease. And we're going to get into this. And on the opposite, vaginal microbiota that lack lactobacillus are basically now recognized as what we call non-optimal, and they tend to be dominated by they lack lactobacillus, and they associated with sexually transmitted infections, prematurity, HPV, progression to cancer. There's a lot of adverse outcomes that are associated with those kind of microbiota. So we've been, over the years, we've been able to basically categorize those different kind of microbiota based on their composition, and we showed that early on, there was broadly 5 major categories, 4 of which are dominated by species of lactobacillus. And we have Lactobacillus crispatus, Lactobacillus iners, Lactobacillus jensenii, and Lactobacillus gasseri, which are the main lactobacillus that we find in the vagina. And there's this community state type 4 that lack lactobacillus and comprises a different conformation of a wide arrays of strict and facultative anaerobes. And now this has been expanded and refined in terms of categorization. But those categories are kind of very interesting because they also represent functionally some major differences, and we try to explore those differences. So one of the pathogens we study in my laboratory is Chlamydia trachomatis, which is the etiological agent of chlamydia, the genital infection, and others. But we looked specifically at the genital infection. And so we looked at women who presented in the clinic with chlamydia. And we simply characterized their microbiota, and we had 160 women. They all ultimately got treated with azithromycin, which is the recommended treatment, 1 gram of azithromycin at 1 dose at diagnosis. And those women were resampled 3 months later. And so what you can see that at time of diagnosis, usually they have pretty strong inflammation, and the microbiota tend to be this CST4, which does not contain lactobacillus. But there's quite a few microbiota that actually do contain lactobacillus, but they contain a lactobacillus that's called Lactobacillus iners. And post-treatment, what we see is that there's a return, if you want, in the population level of that community state type 4 that lack lactobacillus, but there's an expansion compared to the population that's negative and never been infected with chlamydia. You can see there's an expansion of the community state type that contain lactobacillus iners. And this was surprising to us because we would not expect this. It could mean that potentially that those who return to a microbiota they were prior to infection, meaning that this lactobacillus actually could provide an increased risk for the infection. And we wanted to explore this. Obviously, there's also a possibility that Lactobacillus iners could also be resistant to the treatment, the antibiotic treatment, and lead to this pattern that we see. And so we looked at antibiotic, and indeed, Lactobacillus iners, there's about some, not all the strain, but some strain tend to be resistant to azithromycin, as well as Gonorrhea vaginalis, which is the prototypical microbe that's associated with this community state type 4. But On the other hand, what we wanted to explore is why does lactobacillus iners capable of increasing the risk for chlamydia infection? So chlamydia is an intracellular pathogen, so it's somewhat challenging to study because we can't grow it in the lab in a flask like you do with E. coli. You have to infect epithelial cells and look for what we call inclusion, which is basically a lot of different chlamydia particles that are inside the cells that form an inclusion, and they stain in green here on those figures. And you can see the infection on cells that are just grown in different media. It works actually very well. You get a pretty strong infection. If you expose cells in our 3D, we have a 3D biomimetic model of the vaginal epithelium. If you expose those cells to Lactobacillus crispatus, what you see is that we cannot infect chlamydia after exposure to crispatus, to Jensenite. But when we expose it to Lactobacillus iners, the infection actually does proceed. And so one property that Lactobacillus iners has compared to the others is that it does not produce one of the two isomers of lactic acid. So it doesn't produce D-lactic acid, while all the other species of lactobacillus that are found in vagina do produce D-lactate. And so this is the main difference. So we basically hypothesize potentially that D-lactate could actually be a compound that actually provide this protection against chlamydia. And so we measured D-lactate and L-lactate in those culture of crispatus, Jensenite, and iners, and you can see that iners does not make lactic acid. But then we took Lactobacillus iners and we supplemented the culture with D-lactate. And what you can see that when you do this, you actually restore the phenotype of protection and to a lesser extent if you just add L-lactate, but then you have such a high amount of lactic acid that there's some level of protection here as well. So we repeated this at different pH, this time with pure lactic acid, and we didn't include the bacteria. And what we showed here, which it's even more interesting, is that at pH 7, there's no effect. So you still have the compound is present, but the pH is high, there's no effect. But then if you expose the cell first before the pathogen, you have protection. However, if you expose the pathogen to lactic acid prior to infection, you don't have protection. And if you expose both, obviously, you have protection. And so what this indicated to us is that lactic acid was not acting directly on the pathogen, as one might have expected, but it was acting on the host cells to render those cells actually protected against infection. And we wanted to explore this mechanism. And so you can see that, interestingly, if you expose those cells and actually remove lactic acid or actually culture of lactobacillus or supernatant of lactobacillus, what you can see that protection is actually lasts quite a while. So post-exposure, so for 30 minutes you expose the cells, and then you try to infect them 24 hours later or 48 hours later, you can see that actually that protection remains. So there's a kind of a long-lasting effect of that original exposure to lactic acid or supernatant of lactobacillus culture. So this was quite interesting. So one thing we did, we looked at in human sample, in biological sample, in women who have different kind of microbiota, and we looked for features of the host in through microRNA sequencing that could potentially explain this phenotype. And so what we found is that in those biological sample, when you compare different microbiota and expression, what we find is that epithelium cell proliferation was actually differentially affected by the different microbiota. It was increased when you have a microbiota that doesn't have lactobacillus, but it was also reduced quite dramatically when you have a microbiota that's dominated by lactobacillus present. So this is in vivo. So because we know, by definition, that non-optimal microbiota, those that don't have lactobacillus, actually do increase the risk, then we ask the simple question, is cell proliferation required for the infection? And so we blocked the proliferation using inhibitors of proliferation, and you can see that the proliferation is actually blocked. And then you try to infect those cells, and you just cannot. So it just indicated to us that cell proliferation was on the pathway to and certainly critical for successful infection by chlamydia. And so one of the things that we went back to our model, and we did some gene expression this time on the model in the presence of different prototypical lactobacillus or Gardnerella vaginalis, which represented this CST4. And what we found is that there's actually an epigenetic modulation of cell cycling in the vaginal microbiota. And you can see histone deacetylase 4 is actually inhibited by Lactobacillus crispatus, for example, and while the inhibitor of cyclin D1 kinase is actually overexpressed. So when you have a lot of HDAC expressed, it inhibits the inhibitors and allow the cell cycle to proceed. When you don't have histone deacetylase 4, you have increase of the inhibitors, which can then actually inhibit the cell cycle, which actually supported very well a reduction of the cell cycle by lactobacillus over Gardnerella vaginalis. But you can see also that Lactobacillus iners was not able to actually inhibit histone deacetylase the same way that it was than Lactobacillus crispatus. So now we have somewhat of a much better understanding of how the microbiota is affecting host cell in the protection against chlamydia. And in those transcriptomic experiments, we actually had identified other inhibition effect of lactic acid or even lactobacillus on, for example, EGFR, which is critical for the attachment and the basically internalization of chlamydia. But cell proliferation was identified as a major factor. We also had an effect on the estrogen receptor, which is also inhibited by Lactobacillus crispatus and jensenii, which makes sense as it does increase cell proliferation. So we now really starting to develop a much better understanding of how the microbiota can affect host cell in the context of a different microbiota, in the context of sexually transmitted infection. But one thing that was really interesting, I think, in this study is that we always thought that the microbiota would affect the pathogen directly, especially when it comes to lactic acid, low pH. It's always thought that was the mechanism by which this would happen. But our study really started to show that the microbiota affect the host and in shaping it in a way that it's actually resistant to infection. So all those studies, and especially getting into the functional aspect, is really going to help us develop novel and improved strategy to restore optimal protection against sexually transmitted infection, which is a worldwide problems. Chlamydia is the most reported sexually transmitted infection in the US and actually worldwide. So with that, I'd like to thank some of the people who actually, the two people who did a lot of that work that I presented, Vanessa Edwards and Steve Smith, my lab as well, the Microbiome Service Laboratory, which perform a lot of our experimental and sequencing data generation, and some of the tool that we generated. Virgo is a tool that allows you to analyze metagenomic and metatranscriptomic data set from the vaginal microbiota. It's a gene catalog of the vaginal microbiome. So with that, I'll be happy to answer any question.
Thanks a lot, Jacques, for this excellent talk. There's already a question from Gloria that actually can't write in question and answer. So Gloria, do you want to ask directly?
Sure. Jacques, thank you so much. It's great to see your new advances in characterizing the vaginal microbiota. My question is, have you studied the functional aspects of the vaginal microbiome during pregnancy? Do you have any clues on the on-off manifestation of strep B, which leads to so many antibiotic treatments during labor?
Yeah, no, we haven't really studied and looked at strep B in very much detail. It leads to the use of antibiotic, which is definitely a major issue. But in terms of its association, our interest is mostly in prematurity, and the association with prematurity is not as strong as the association of the rest of the microbiota with prematurity. So I cannot really answer that question. In terms of function of the microbiota in pregnancy, that's some ongoing work, and we don't really have any data to present yet.
And there's a second quick question from Joanna Rolo that asks, what would be the role of Lactobacillus iners in the vaginal microbiome? It looks like a pathogen.
That's a very good question. Lactobacillus iners is a very strange lactobacillus. It has a genome that's about half the size of the other. It actually has the ability to produce a toxin that's a cytolysin. So the thing, we have a preprint that you can go check out where we looked at metatranscriptomic analysis of different microbiota. And Lactobacillus iners is very interesting. So for example, one of the things with function that's very, I think, fascinating is that and people should consider, is that it can vary by context. So a microbe can be present in a microbiome, but if the partners that are with it are different, that microbe is going to do things that are different. And Lactobacillus iners is exactly that. When it's present with other lactobacillus, that cytolysin is never expressed. When it's present with Gonorrhea and other anaerobes, that cytolysin is being expressed. So presence, absence, or even presence of functions is one way to look at things, but you also missing a part of that picture. And understanding expression and context, I think, is really critical to understanding the beneficial and adverse role of the microbiota. So it's true that iners could be a problem, but it seems to be a problem when it's present with microbes that are, the one that you don't want to see in the microbiota.